to our next speaker, which is Sacha van Albada. So please, Sacha, come to the scene. So Sacha van Albada is presenting large-scale spiking neuronal network models, and she is continuing, I would say, the talk of Hamid that we heard, that he tried already to connect anatomy and physiology. And of course, these are two sides of one and the same medal, and we are very curious to see, Sacha, how you will present connectivity in your talk.
Good afternoon, everyone. My group develops large-scale spiking network models of the cerebral cortex of monkeys and humans, investigating under what conditions realistic resting state dynamics arises at multiple scales. The main question we are trying to answer is, how does the complex neural network structure of cerebral cortex give rise to its dynamics on different scales, from the microscopic spiking activity to the macroscopic interactions between areas? At the top, you see the parallel spiking activity of 140 neurons in the primary visual cortex of a lightly anesthetized macaque monkey. And you see that the population activity displays multiple timescales. The bottom figure visualizes the well-known phenomenon of resting state networks in fMRI, consisting of groups of areas that co-activate on timescales on the order of 10 seconds. Another aspect of cortical resting state dynamics is that the firing rates of neurons differ across layers, tending to be highest in layer 5 and lowest in layers 2 and 3. And during brain states akin to the resting state, activity tends to propagate down cortical hierarchies, as illustrated by this EEG study on the bottom right, comparing visual imagery with visual perception. In the perception condition, subjects watched short movies or imagined, and during the imagery condition, they mentally replayed those movies or imagined riding a magic bike without pedaling to a destination of choice. During the imagery condition, which is more similar to the resting state, there was more feedback, Granger causality, from the superior parietal lobule to the inferior occipital gyrus. And these are some aspects of cortical resting state activity we tried to reproduce in our models. As a first model system, we chose macaque vision-related cortex because of the richness of the available data sets and because it forms a stepping stone to the human brain. Another advantage is that it's organized in a more or less regular manner, as exemplified by visual hierarchies. This allows us to make informed guesses about connectivity where the experimental data are lacking. The vision-related areas in macaque cortex contain about 800 million neurons per hemisphere and can be parcellated into 32 areas, which are not all purely visual, but somehow involved in vision. This includes occipital areas like primary and secondary visual cortices, temporal and parietal areas involved in motion processing and object recognition, but also prefrontal and parahippocampal areas involved in eye movements, attention, memory encoding, and retrieval. Because 800 million neurons are still too many to simulate routinely, we start by representing each area by a square millimeter microcircuit with the full density of neurons and synapses, leading to a total of about 4 million neurons connected via 24 billion synapses. This network is still very large, so we run it on a supercomputer in Jülich using the NEST simulator. We model each area by a microcircuit similar to this one, using leaky integrated fire neurons, divided into 8 populations, 2 in each of the 4 layers shown. And to focus on the influence of the connectivity on the dynamics, we represent each neuron with the same parameters, with the same intrinsic dynamics. And the connectivity is population specific, given by connection probabilities, but otherwise random. So connection probabilities that are specific to the source and target populations. And an external Poisson drive represents the non-modeled parts of the brain. The model produces asynchronous irregular spiking activity like shown on the right, with layer and population firing rates matching experimental data. Coming back to the notion of visual hierarchies, depending on their relative hierarchical positions, vision-related areas have different laminar connection patterns, as most of you will know. So feed-forward projections originate mainly in the supragranular layers 2 and 3 and terminate in layer 4 of the target area. Feedback projections originate mainly in the infragranular layers and terminate outside layer 4. And for areas at the same hierarchical level, projections tend to originate approximately equally in supra and infragranular layers and terminate in a columnar fashion across all layers. One can then define a hierarchy that is as consistent as possible with all the pairwise connection patterns. However, due to deviations from the stereotypical laminar patterns, there's some indeterminacy in the positions of the individual areas in the hierarchy. And one way of resolving this indeterminacy is to define hierarchies based on the local cytoarchitecture of areas. So-called architectural types characterize the neuron density and the distinctiveness of the lamination of cortical areas. Primary visual cortex, area V1, is a eulaminate area with distinctive layers, a high neuron density, and a thick layer 4. And going up the hierarchy, the neuron density decreases and layer 4 becomes thinner, eventually disappearing altogether in agranular areas. As opposed to neuron density, the volume density of synapses remains roughly constant, so that in higher areas, the neurons receive more synapses per neuron. Architectural types are relevant for us because we need to determine the population sizes for a model. And specifically for areas for which neuron densities were not measured, we can estimate these from the architectural types. And we then multiply neuron densities with the total cortical thickness and the relative laminar thicknesses in each area to compute the number of neurons in a given area and layer. Next, we need to determine the connectivity between areas. And for this, we use axonal tracing data collected in the COCOMAC database and from the lab of Henry Kennedy. The COCOMAC data are mainly qualitative, telling us which area is connected to which, and the Markov and Kennedy data are quantitative, measuring the fractions of labeled neurons, or FLN, in each area, sending connections to the area injected with the retrograde tracer. These data are extensive, but still incomplete, so that we need statistical regularities to estimate the missing values. And one thing we use is that the connectivity decays approximately exponentially with the distance between areas. According to the currently available data, about 2/3 of the possible connections exist. But what's more important is that the density of connections varies widely, spanning about 6 orders of magnitude, which we need to take into account. As a sanity check, we tested whether areas that perform similar functions are more strongly connected in our model. And for this, we used a clustering method called the map equation method, in which a random walker travels across the network with a probability proportional to the connection probabilities, and a Huffman code description, that is like the country and area codes of a phone book, is derived according to how often the random walker visits the nodes. And the areas with the same first few digits of their phone number then cluster together. This reveals that primary and secondary visual cortex form a small cluster. The areas of the ventral stream on the left, which perform object recognition, cluster together. The areas of the dorsal stream, which process moving stimuli, cluster together on the right, and also the frontal areas and superior temporal polysensory areas. And finally, we have a cluster of areas, the blue one in the middle, for which the connectivity information is relatively incomplete, so that this cluster has to be taken with a grain of salt. But overall, this community structure is very reasonable. So far, we have considered the area level connectivity, but we still have the cortical layers to worry about. And some quantitative information about laminar connection patterns is available in the form of fractions of supragranular labeled neurons, or SLN, from retrograde tracing studies. SLN measures the proportion of labeled cells in layers 2 and 3 relative to the cells labeled across all layers. Because the data are not available for all pairs of areas, we again estimate the missing values using a statistical fit, this time against a logarithm of the ratio of neuron densities of the pair of areas involved. When the target area has a higher neuron density than the source area, this ratio is positive and the connection goes down the hierarchy, emanating mostly from the lower cortical layers, and vice versa for the connections going up the hierarchy, which emanate mostly from the upper cortical layers. And for the target side, we also have some information about the laminar patterns. Some of this comes from anterograde tracing studies collected in the COCOMAC database, and the rest we can guess, because a given laminar source pattern is associated with a given laminar target pattern. These target patterns describe the location of the synapses rather than of the cell bodies of the target neurons, which may be in a different layer than where they receive the synapses. So to associate synaptic locations with the locations of the target cell bodies, we use morphological reconstructions of neurons from cat primary visual cortex, as it was the only comprehensive layer-specific data set available. And we then assume that the probability for a synapse to be established on a given type of neuron is proportional to the total length of its dendritic tree in the given layer. And then putting together the area and layer-specific connectivity, we arrive at a connectivity map for all the areas and layers in our model, shown on the bottom right. Simply putting together the connectivity directly from the data, it's difficult to obtain reasonable activity in such a complex model. Therefore, we use mean field theory to find out how to change the connectivity slightly to obtain a more realistic ground state. However, this ground state still looks very much like our isolated microcircuit model without the slow fluctuations seen in the experiment. And to remedy this, we gradually increase the synaptic strengths of cortical-cortical connections to evoke inter-area interactions as parameterized by this variable chi. And it turns out that we have to increase the synaptic strengths onto inhibitory neurons more than onto excitatory neurons in order to remain in a low rate asynchronous irregular state. And there's a sweet spot at chi is 1.9, where the activity shows the same mix of short and long fluctuations that is present in the experimental data. The network needs to be poised just below this transition between a state with low rates and one with high rates. And this corresponds to a well-known phenomenon of dynamical slowing when a dynamical system approaches an instability. And this type of activity with fluctuations on multiple timescales does not occur in a model of a single microcircuit, and it's an effect of the multiple microcircuits being connected together. The resulting activity looks like this for 3 example areas out of the 32 areas in the model. Primary and secondary visual cortices and the frontal eye field, FEF. And the spikes of the excitatory neurons are shown in blue and those of the inhibitory neurons in red. The spike rates and activity correlation between neurons are generally low, like in the brain. And the irregularity of spiking of individual neurons is close to that of a Poisson process, so which is also realistic for cortical activity. And the average rates of each area shown at the bottom fluctuate on short and long timescales. We assessed the agreement between the simulated and experimental data from primary visual cortex. The experimental data contain intervals with less and more synchrony, for which the spectra are given by this green and purple curves. But overall, the spectrum of the activity looks like the yellow curve, and from the simulation like the black one, which fits quite well apart from this little bump between 20 and 40 hertz. And we also looked at the distribution of spike rates across neurons shown at the bottom right. So computing the average rate of each neuron and then considering the distribution of these average rates. The yellow curve for the experiment is a bit more difficult to see because it matches the black curve from the simulation so well. So this good match occurs when the network is poised just below the transition between the low and high activity states I showed before. And when the inter-area synapses are chosen to be weaker or stronger, the simulations don't match the data. So we have to be near this instability. We also looked at the order in which the activity propagates across areas, analogously to the hierarchy based on structural connection patterns. We determined a hierarchy based on pairwise orderings of the activations of the areas. That is, we look at the correlation function for each pair of areas and see whether the peak occurs at a positive or a negative lag, indicating which of the two areas tends to be activated first during the ongoing activity. And then we ordered all the areas in such a way as to minimize the number of violations of the pairwise orderings to obtain a unique sequence in which areas tended to be activated. It turns out that parietal areas are activated first, followed by temporal and occipital areas. And finally, a reduction instead of an increase in activity occurs in frontal regions. And this ordering of parietal before occipital areas corresponds to the feedback propagation observed in the EEG experiment that I mentioned in the beginning.
15 minutes
Finally,
are over. 15 minutes are over.
Oh, okay. Yeah, what would you like me to do? I can, because I haven't talked about the human model, but it's much briefer
Please
than.
take 2 more minutes, but somehow we have to.
Okay.
Yeah.
Yeah. So the functional connectivity, we compare the correlations between the simulated synaptic inputs to each area with functional connectivity between the areas from macaque fMRI. And the comparison reveals that the match peaks around the same strength of inter-area synapses where the microscopic spiking activity matched the experimental data, and which is as good as one can expect based on the variability between individual monkeys. Okay, and the code is available on GitHub. And recently in a collaboration also with Timo and Klaus Hilgetag, we have started to work on an analogous model of human cortex. We don't restrict ourselves to the vision-related areas here, but model an entire hemisphere, but in a coarse parcellation where only 34 areas are distinguished. And that yields a model size comparable to the macaque model, but more synapses, because human neurons have more synapses per neuron. And we take the laminar thicknesses from the BigBrain, and the neuron densities are still mainly from von Economo and Koskinas, but we are gradually replacing them by the more precise estimates from Timo and from the BigBrain. The area level connectivity is from DTI, from the Human Connectome project. We determine the laminar patterns using predictive connectomics, using the regularities observed in macaques. So depending on the relative neuron densities of the different areas. And we do this mapping again of the synapses to the target cell bodies and have an intrinsic connectivity that is scaled version of the Potjans-Diesmann model. We again have a reasonable community structure and use mean field theory to find reasonable parameters, which suggests a very strong role of layer 5 for ensuring low rate asynchronous irregular activity. And then first simulations show that the mean field theory is able to find reasonable parameters. And yeah, we're starting to look into this, into the functional connectivity, and we'll continue to investigate. And with that, I thank you for your attention.